Hello all, I am Priyadharshini, Assistant Professor, Department of Biotechnology, JJ College of Arts and Science, Pudukkottai. Today we are going to discuss about the biological databases, and it's a overview of the databases. Here the introduction about the biological database. Database is an organized collection of data. Here, the data's bioinformatics is concerned. The data are in the form of DNA sequences, protein sequences, and the RNA sequences. It can be assessed, added, retried, manipulated, and also be modified. A database is for storage, and we can manage, connect, and distribute the data all over the world so that the scientist can able to retrieve the data easily, and the sets of the data are programmed into a software that manages the data called the DBMS, that is database management system. And here are the classification of data bases. First one is primary, secondary, and the composite databases. Here the classification is based on the type of data. That is, in the primary database, we can get the raw sequences, that is the original sequences as such recovered from the sequences. In case of primary data, here the informations are from the primary data. The secondary database informations are from the primary database that can be processed and retrieved, and the raw data banks is which is available in the primary database. That is, example for the primary database is ProSite, Prints, Blocks, PFAMs, Identify, etc. And the composite data banks. The composite data banks, it can collect and present the data after comparing, filtering them from different primary, and exhibit only the non-redundant sequences. That's composite databases. Primary database that consists of nucleic acid database, that is GenBank, EMBL, DDBJ, and protein sequence databases, Swiss PIR, SwissProt, and SwissProt in another form is the UniProt. Protein structural data basis, PDB, and scope cath, that things will also come under protein structure database. And the metabolic database example is KEG, Kyoto Encyclopedia of Genes and Genomes. Nucleotide sequence data bank, it composed of nucleic acid sequence entries. Here, the data repositories that accepts only the nucleic acid sequence data bank, and it can be freely available to all the scientists all over the world. It will minimize the work of the scientist. Why? Because each and every places they are sequencing a particular genome, means it is a time consuming. So if one person sequence the genome, that is entered into the data bank that can be retried by all the scientists over the world. GenBank, EMBL, DDBJ are the primary nucleotide sequence data banks. All the three of the data banks, they can interchange their data on the daily basis. So we can get the sequence from all the three data banks. And this is a GenBank of NCBI, hosted by National Center for Biotechnological Informations. It is situated in the campus of US National Institute of Health, USA. It also offers publicly available nucleotide sequences, and offers direct submissions of sequence by the author itself, and it is a user friendly. It can able to access by using the entrance retrieval system. And this is the home page for GenBank. See, in the search, we have it has some pull down button. We have to select an option interest, and for here we have to we have to type the name of the organism. And the DNA Data Bank of Japan, DDBJ, it started in 1986, hosted by National Institute of Genetics Japan, gather data primarily from the scientists in Japan and other researchers all over the world. Why? Because it is maintained by the Japanese. And here the DDBJ is concerned, it gives us an internationally recognized accession number. Not only the DDBJ, all the data banks will also contain some accession number, and it can able to manage and developing tools for the analysis and the visualization of the biological data banks, and also conducting courses for the beginners to reduce the complexity of the biological data analysis. And this is the DDBJ homepage. See, likewise in the GenBank, the same information we can retry from the DDBJ also. And EMBL, European Molecular Biology Laboratory, established in 1974, hosted by the European Bioinformatics Institute. It is a non-profit as such from the EMBL likewise. The sequence that are stored in the database would be expressed in the biological states, and the stored data corresponds to wild type sequence without mutation or genetic manipulation. And this is the home page for EMBL. And next one is a protein sequence database, SwissProt, PIR, and UniProt. UniProt is the most user friendly to get the sequences from the data bank. And this is information about the SwissProt. SwissProt is from Swiss Institute of Bioinformatics and European Bioinformatics Institute. It has highly quality and manually annotated protein sequences. It determines post-translational modifications also. It consists of two sections, UniProt KB and SwissProt KB. And this is the SwissProt homepage. From this itself we can go for UniProt also. And next one is PIR, is a primarily protein sequence data banks established in 1984 by National Biomedical Research Institute, that is NBRF, and it also assisted the researchers. All the data banks can consist of the same information. This is the home page for PIR. And this is the UniProt, same as that of the SwissProt. Components of UniProt is UniProt knowledge base, reference clusters, archive, metagenomic and environmental sequences. And this is the page for UniProt. And protein structural data bank, it contains the structure of the tertiary structure of the protein sequences. This similar protein exhibits changes in primary, secondary, tertiary, and also the quaternary structures. And PDB is a specialized data bank for the storage of the protein structures. It is maintained collaboratively by the Structural Bioinformatics Institute, RCSB. The data can be obtained by X-ray crystallography, NMR, and are submitted to the PDB. And this is the home page for PDB. And this is a model organisms database, means it is specific for the particular organism. SGD means it is for Saccharomyces alone. AGR means Arabidopsis thaliana. HGP means only for human. RGDB means only for rat. And this is the biodiversity data bank. And thank you so much for watching.